My name is Michael Greeks. I am the digital marketer here at Slingshot Biosciences, and very excited to introduce you to Swetha, who's really going to go through some of the trials and tribulations that are related to or associated with high parameter cytometry. So we're going to talk to you today about SpectraComp and ViaComp controls, or as we like to consider them, the dynamic duo when it comes to decreasing assay variability, reducing complexity, and improving accuracy. So with that, let me hand it over to Swetha.
Thank you, Michael. Thank you, everyone, for joining the webinar today. I'm a scientist at Slingshot Biosciences, and today I will be talking about how to simplify your compensation and unmixing in high-parameter cytometry using SpectraComp and ViaComp. So before we get started, this is a semi-interactive webinar, so we will be having few poll questions throughout the presentation. And anytime during the presentation, if you have any question, feel free to put them in the Q&A panel, and I will address them at the end of this presentation. Let's look at the agenda. So I'll be giving a short introduction to Slingshot Bio, and then I'll talk about the challenges that arise in high-parameter cytometry. Then I'll walk you through the importance of compensation and unmixing, then talk a little bit about SpectraComp and ViaComp and how it can be used in the 9-color TBNK panel, as well as go over some case studies from external resources, like a 40-color panel from Cytometry Biosciences and a 30-color panel from BMS. So for those of you who don't know who we are, Slingshot Biosciences is a startup company founded in 2012. We are based in Emeryville, California, and we are focused on developing cell mimics for biological assays. So cell mimics are their hydrogel-based particles designed to behave similarly as that of cells in flow cytometry and other analytical platforms. Over the past several years, cell mimics were used to develop a range of controls and reference materials that support different stages of flow cytometry workflow. So now let's look at those workflows. So this is a brief overview of a flow cytometry workflow. So we have instrument setup, then we have panel setup. Then after the panel setup is done, you either do your assay development or run an established assay. And if you are a CRO or a big biotech or pharmaceutical companies that is managing multiple sites, then you are also worried about site harmonization or standardization. So controls are essential at every part of this flow cytometry workflow. And different controls are needed at different stages of this workflow, right? You cannot use the calibration beads that you use to set up the instrument for your panel setup. Panel setup might require different kinds of controls. At Slingshot Biosciences, we developed off-the-shelf controls that are designed to support different aspects of this flow cytometry. So we have flow sites for instrument setup. We have scatter bridge for instrument site harmonization or standardization. Then we have a variety of compensation products for your panel setup. And then we also have several biomarker controls that can be used as an assay control or a gating control while running your assays. And apart from these, we also have antigen quantitative controls. So before we go into the details of high parameter cytometry, let's go to our poll question 1. What type of controls are you currently using in your flow cytometry experiment? Okay, I'm going to end the poll now and share the results. So I see most of you have selected compensation and unmixing. And to be honest, if you ask me, I would say all of these controls are important for flow cytometry workflow. But with growing complexity in your panels, I would say selecting right compensation and unmixing control can have a very huge influence on your data. So let's see how. Okay, so we are currently in the era of high parameter cytometry. So flow cytometry has evolved a lot over the decade. So initially, we were only analyzing few markers. Now we are at a stage where we are able to analyze over a dozen markers in a single cell. And with modern spectral cytometers, we can measure over 40 parameters simultaneously in an individual cell. That is enabling us to perform all these applications. So I want to show you the complexity of the data analysis for a 40-color panel using a paper that I found. This is the OMIP 69. And here you can see with the increased dimensionality, there is also an increase in complexity. So let's see that visually. So early cytometry panels only have 3 to 6 markers, and they were fairly simple. And then with modern cytometry panels, we can measure from somewhere between 15 to 20 markers, slightly adding to the complexity. Now, with spectral cytometry panels, we can measure over 40 markers, drastically increasing the complexity. So as the fluorochromes increases, the complexity is also increasing. So this slide reiterates the same, but here I am comparing a 6-color panel with a 40-color panel. So on your left, you can see that the 6-color panel, I've chosen the fluorochromes that are spaced apart and have minimal spectral overlap, spillover interactions, leading to the lower sensitivity to compensation and unmixing. Now, on your right side, you can see that this is a 40-color panel, and you see there are many fluorochromes, and there is extensive spectral overlap, leading to numerous spillover interactions. So that can lead to higher sensitivity. So this means that small differences in reference control or spectral signature can significantly affect unmixing accuracy, which in turn can affect the assay reproducibility. That's why compensation and unmixing becomes very important. So what happens if you have poor compensation? Poor compensation or unmixing can make your bright signal bleed into other detectors, causing spreading errors. And now with the dim population, it gets harder to resolve, so you have poor separation. And small errors can also cause false double positive populations. So this means even small compensation errors can lead to incorrect biological conclusions. So let's look at how an uncompensated sample looks versus how a compensated sample looks. So on the left side, I'm showing an uncompensated sample. Here, I'm plotting the data against CD8 and CD4. So you can see CD8 is in NXO4 700 and CD4 is in Cy7. You can see when the compensation is not applied that the CD8 is highly undercompensated and it spills over into CD4. But for the same sample, when I apply compensation, you can see the spillover, CD8 doesn't spill over into CD4 anymore. And now you have a clean CD4 and CD8 populations. So this shows that compensation is not changing any underlying biology. It is only correcting the fluorescence spillover between the detectors. So these are some of the common reference controls that are available. The industry standard cell-based controls, which match the cell autofluorescence, obviously, but it is often limited by brightness and availability. On the other hand, you have commercially available beads. They are bright and consistent, but they do not match the cell scatter profiles of the cells, and they also do not replicate the autofluorescence, and they have their own practical limitations. Before I show our solution to this limitation, let's go to the poll 2. What type of controls do you currently use for your compensation and unmixing? Okay, so to address the limitations that I was talking before, our solution is to use SpectraComp and ViaComp. So SpectraComp is nothing but an antibody-based compensation control, which is very similar to cell properties. It is decorated with capture antibodies and is used to provide a clear resolution of positive and negative populations on the biomarkers. ViaComp is a viability control that distinguishes live cells from dead cells, and it replaces the need for heat-killed cells, saving time and resources. So these cell mimics are designed to combine consistency and brightness of the beads with cell-like characteristics. Now let's look at them individually. SpectraComp, like I said, it is an antibody-based compensation control. It can be used as a single-stain controls for individual antibodies to set gains for compensation and unmixing. So SpectraComp is compatible with 3 host species, mouse, hamster, and rat. And if you are working with rabbit and human, we also have a product for that, SpectraComp XD. Then we also have a product called SpectraComp Flex, where you have an opportunity to use positive and negative mimics separately. Now, ViaComp is a viability control, just like I told before, and it has scatter very close to cells. That is very similar to cells. And it is compatible with both DNA dyes and amine dyes. So it also replaces the need for generating dead cells using either heat killing or chemical treatments. So the one advantage of ViaComp is that you can use this as a process control as well as a compensation control. Let's look into some data, right? Before I show some data, let's look at the critical requirements for compensation and unmixing. There are many rules for compensation and unmixing. I'm not going to go over all the rules, but I want you to focus on the top 3 rules. So the first one is that control should be as bright or brighter than samples, or it should include a positive and negative populations well separated. And then the third one is that the autofluorescence should match the sample. So let's see if SpectraComp and ViaComp are complying to these requirements. So requirement 1 and 2 is that it should have bright signals and it should have a clear separation. So on the right side, I'm showing the separation for a CD16 marker, and you can see that it has a very clear negative and positive separation. And on the right side, I am comparing SpectraComp with NK mimics, which are usually dimmer and less in frequency. And you can see that SpectraComp cell mimics are very bright compared to those cells. Now, looking at the third requirement, which is cell-like autofluorescence. Here, I'm showing an overlay of cell mimics, cells, and commercially available beads. You can see that the cell mimics, which is in orange, is very similar in profile to that of cells, which is in blue. But the beads have a significantly different fluorescence profile than that of cells. So ViaComp is very close to very similar to SpectraComp, and it has autofluorescence very similar to cells as well. Now let's look at the brightness for a ViaComp. So here I'm showing the data for both DNA dyes and amine dyes, and you can see that the ViaComp is as bright as cells. Traditionally, in most of the labs, people tend to generate dead cells using either chemical treatments or ethanol or heat killing. So one benefit to ViaComp is that apart from matching the scatter, it also replaces the need for generating the heat-killed cells, serving as a better alternative for cell-based control. So now this leads us to the last poll question of the day. What are you typically using to generate dead cell controls for your viability dyes? Okay. So I see a lot of you are using heat kill cells. And some of you said you skip your viability controls. I would say don't do that. Viability control is very essential. But for those of you who has selected heat kill cells, like I said before, ViaComp has an advantage to replace the necessity to heat kill your cells. I've shown the data individually for SpectraComp and ViaComp. Now let's look at them in a compensation or unmixing to see if it provides good compensation or unmixing accuracy in a multicolor panel. So here I'm comparing the industry standard cells with commercially available beads and our cell mimics. So in this experiment, I've used SpectraComp as an antibody-based compensation control and ViaComp as a viability control. And I've performed a 9-color TBNK panel on Cytec Aurora. So on the right side, I'm showing you the overlay of all the dot plots generated using either beads, PBMCs, or SpectraComp and ViaComp. Now let's look at them closely. I will start with scatter. So here I am comparing SpectraComp with biological cells and beads on the left side. And I'm comparing the side scatter and forward scatter for the same for ViaComp on the right side. You can see that the beads are in yellow. So for the sake of comparison, what I've done for this experiment is that I kept the grade very broad and same across all three controls. And I was trying to eliminate the debris while gating for the biological sample. So while doing so, you can see that most of my beads are not falling within the gate. But SpectraComp has a very similar scatter profile as that of biological cells. So oftentimes we tend to use a threshold for our flow analysis. And in that cases, then you would have to adjust your gains to make sure that the beads are falling within the axes. So let's look at ViaComp. The ViaComp is very similar to SpectraComp, right? It has a scatter profile very similar to cells. And you can see that the commercially available beads are not even very close to that of cells or ViaComp. And now looking at the fluorescence intensity for both SpectraComp and ViaComp. And I'm on the right side, I'm comparing fluorescent intensity of SpectraComp with biological cells or beads on CD3. And you can see for both SpectraComp and ViaComp. And in this particular experiment, they're very brighter compared to the beads or biological cells. So in this slide, I'm showing the dot plots for live dead NIR, compensated with cells, which is on the leftmost, then compensated with ViaComp in the middle, and then compensated with bead on the rightmost. So let's start with cells. Here you can see that the live percentage is around 80%, but you can see that when I used cells for compensation, there is a slight spreading occurring in the live dead, which is not impacting the percent population though. Now let's look at compensation with what happens when it's compensated with the beads. So in this plot, I'm trying to maintain the gates across all three controls, right? So you can see that there is a huge unmixing error when I'm using beads, and the live population is separated into two populations. And when I'm maintaining the gate, I am seeing a reduction in my percent live by 15%. So that's where ViaComp comes in. So ViaComp has given a very clean and similar profile to that of cells, maintaining that live percent very similar to the cells. So sometimes these plots not only have impact on themselves, but it also has impact on other markers. So let's see how this live dead is impacting the other markers. So just like before, I am comparing the all three controls. Here on the left is the compensated with cells, then is compensated with beads. And since I am using the biomarkers, I'm showing the compensation with SpectraComp in the middle. If you notice at first glance, I don't think I see any major, visually, I don't see any major differences. When I look at it very closely, the CD8% populations for cells and SpectraComp are, the CD8 is around 30% and CD4 is around 55%. And the 4 to 8 ratio is very close to each other. But when you look at the beat, there is a slight overcompensation on CD8. And you can also see that it has impacted the percent population of CD8s and 4s. Here, the CD8 has increased to 50% and the CD4s has reduced to 30%. So this tells you that choosing the right control becomes very important. Okay. So till now, I've shown the data generated in a 9-color panel, but now let's move to a high parameter cytometry panels. So I am showing case studies. So this is case study 1. This is from Cytec Biosciences. They have published a paper on a 40-color panel. So in this panel, they were comparing the commercially available beads with cells and comparing the spectral differences between them. So the supplemental session of this paper has a lot of data for different fluorochromes and the differences that they see between beads and cells and its impact on unmixing inaccuracies. But here I'm only showing the data for a particular fluorochrome, which is BUV 661, where SpectraComp had huge, but you can see, but let's walk through what is in the slide. So when they have performed compensation with beads for CD39 in BUV 661, they have seen that BUV 661 spills over into BUV 395 and the APC channel. And you can also see that there is a slight overcompensation with BUV 661. The same was shown using a spectral profile on the right. So now let's look at the data that we generated internally. So here I'm showing the compensation with cells and then compensation with SpectraComp. And on the right, I'm showing compensation with two kinds of commercially available polystyrene beads. So you can clearly see that when I'm using SpectraComp, it is not showing any unmixing inaccuracies, and it is matching very closely to that of cells. But when I am using polystyrene beads, you can see that there is a unmixing, it is introducing some unmixing artifacts, and which can heavily influence the data quality. So this is a 30-color panel published by Bristol BMS, which many of you know that it is a very big pharmaceutical company that is focusing on immunotherapy research. So they've published a 30-color panel where they were comparing cells, UltraComp, and SpectraComp. So now let's look at their panel design. So this panel has around 29 markers with a viability die and 6 open channels so that they can do 6 drop-in markers. To quantify this comparison, they have used similarity index, and they're comparing the similarity index between SpectraComp versus cells and then ViaComp versus cells. And you can clearly see that SpectraComp is more closely referencing to the cell spectra than that of UltraComp for most markers. Let's look at the dot plots. So here are 4 dot plots. I'm going to talk about CD45 array. So here you can see that they've compared UltraComp, SpectraComp, and cells. So with UltraComp, which is on the left, you can see that there is a slight overcompensation. And when you look at the cells, you can see that there is a slight undercompensation. But when you use SpectraComp, it is removing all the unmixing inaccuracies and providing a good separation for CD45 array and CD16. So for rest of the markers, you can see that either SpectraComp is very similar to cells or performing slightly better than cells. So they have concluded this study saying that SpectraComp is interchangeable with cells for 23 out of 30 markers. And additionally, SpectraComp also reduced a lot of background noise for many of the markers. So in conclusion, they've said that the SpectraComp has a very similar spectral profile to cells than the traditional beads. Okay, key takeaways. I want to reiterate saying that accurate reference controls are essential for reliable multicolor cytometry. So, to summarize, cell based controls are often limited by brightness or sometimes show variable separation. So, that's where cell mimics come in. They provide a brighter signal and consistent separation, just like cell like characteristics. So, with that, I hope it gives you some useful perspective on how reference controls can influence compensation and spectral unmixing, especially when a panel becomes more complex. And if you are working with a high parameter panels, small differences in the controls can have surprisingly large impact on your final data quality. So, choose your controls wisely. So, if you are planning to attend any of these conferences, just stop by to attend our presentation. So thank you all for joining, and I would be happy to take any questions.
And if you have questions, please feel free to enter them in the Q&A panel. And I see we do have one question come through, Swetha.
Ye-
First question, can these controls be used with different cytometers?
Yes, so most of our controls are suitable on many cytometers. We have tested it on many cytometers. The only cytometer we have heard that there was a problem with was Navios.
Another question's come through is, I work with canine PBMCs and feline PBMCs. Will cell mimics work with that?
Yes, they should. And they don't, please reach out to us and we can make custom products as well.
Do SpectraComp controls work with all fluorochromes?
Yes, they do for work with many fluorochromes. We have tested them internally as well.
Excellent. And then I'm also seeing, do I need to adjust instrument settings when using cell mimics?
So in most cases, no, you don't have to adjust your gain settings because the SpectraComp and ViaComp has scattered very similar to cells. But if you are drawing a very tight grid across your around your cells, then I would say that you might have to do a minor adjustment with your gain settings.
And then another question for you, can these controls be fixed or stored?
They can be fixed and stored at 4C for up to 3 days. So I would be a little cautious beyond 3 days because then you might see a loss in signal intensity. That is correct. We have recreated the panel internally and we used different controls to do the unmixing.
And then next question, do you need to titrate your antibodies? With UltraComp preparing the beads, it's simple with 1 microliter of antibody with beads.
Yeah, so most of the time, what I do is I use the titrations that I use for my cells on my beads as well. But sometimes the beads can be very bright, so you might have to titrate it down.
One question's coming in is, how do we order? Do we just add our antibodies to the mimics?
Yes, you just add your just the way you do it with cells.
And then another question for you, for someone who's starting to work with cell mimics, what would you suggest if this is their first time?
So I would say treat them same as your samples. And we do provide TDSs on our website. So if you need any technical data or protocols, it is on our website. But just treat them as your cells, the way you treat your cells.
Got it. Oh, actually I do. Do you have so in order to do you have a web store or do we have to order over email if we're interested?
I think you can do it both ways. We do have a website that where you can place orders, or you can send it to our info at Slingshot. So you can do it either way.
Excellent. And then I know you had mentioned, like, you showed the BMS case study as well and the SciTech case study.
Yeah, so we have a space on our website where we have resources. Where it's called resources session, where we have all the published papers, articles, and application notes on there. So I would say check that space out.
Perfect. And believe that is it for all the questions. Swetha, I want to take a moment to thank you and also to thank all our attendees. We'll also be sending the on-demand, the webinar recording with additional resources to all of you so that you can have. And if any additional questions come through after the webinar, please feel free to reach out to slingshotbio.com to our support, and we'll make sure that we get those questions for you answered.
Okay.
So,
Thank you. Thank you.
yeah, take care, and everyone, thank you again. Please have a good day.